So a little review here on DNA structure and replication. My biggest concern is especially on eukaryotic stuff, since we're going to be focusing on cancer from here on out. So I'm going to do a quick review on basic structure, which I expect from 1B. You already know basic structure and replication of DNA. My experience, however, has been that you learn a lot about prokaryotes and not a lot about eukaryotes. And there are some differences that make a difference when we're talking about cancer. So to kind of start with nuts and bolts here, you guys know terms like nucleoside and nucleotide. A nucleoside is a 5-carbon sugar attached to a nitrogenous base. That's a nucleoside. A nucleotide is when you add on the phosphate groups. Right. These are the building blocks for DNA. So nucleotides are the building blocks to make DNA and RNA, just like amino acids are the building blocks that make proteins. So a nucleotide includes the 5-carbon sugar, which could be ribose or deoxyribose, and then a nitrogenous base and your phosphate groups make up that nucleotide structure. Nucleotides, again, are those building blocks of DNA and RNA. So they go through different examples here of how you can name nucleotides. I'd rather not get into the nitty-gritty, but it is actually quite obvious, really, because you know with DNA we're talking about deoxyribose, right, is the sugar backbone here. And they name them according to which nitrogenous base you have. They name them according to how many phosphate groups you have. So for example, deoxyadenosine monophosphate. That means deoxyribose is your sugar. Adenosine is adenine, is your nitrogenous base, and there's only one phosphate group. So they name them in sort of a predictable way. When we're talking about the building blocks of DNA, of course, we're talking about deoxynucleotides. If we're talking about the building blocks of RNA, we're talking about what's the sugar? Is it deoxyribose? What does DNA stand for? Deoxyribonucleic acid. What does RNA stand for? Ribonucleic acid. So if the sugar for DNA is deoxyribose, the sugar for RNA is ribose. Okay. So it's actually very similar, almost identical structure. The difference is whether or not you have a particular hydroxyl group between ribose and deoxyribose. So what I expect here is that you know the term nucleotide, you know what a nucleotide is, you recognize that as the building blocks for DNA and RNA that make up that structure. Now, to put the nucleotides together and forming DNA or RNA, right, nucleic acids, so nucleic acid is DNA and RNA. The nucleotides are attached together through a phosphodiester bond, a bond here between the two nucleotides. Interesting thing to note, what you see here is the sugar group of one nucleotide is attached to the phosphate group of the next nucleotide. Right? So I see some people nodding and some people, not sure, I've seen this before. So the sugar group of one nucleotide is attached to the phosphate group of the next nucleotide. That's how nucleotides are attached together. That sugar group, whether it's ribose or deoxyribose, to the phosphate group of the next nucleotide. What that does is create an alternating phosphate sugar backbone. So here's our first nucleotide. We see the phosphate group of this nucleotide. Here's the sugar group of this nucleotide. Here's the nitrogenous base of this nucleotide. Attached to the next one here, it's attached to the phosphate group of the following nucleotide, which means you wind up with a structure that's phosphate, sugar, phosphate, sugar, phosphate. It's alternating phosphate sugar groups that form the backbone of a nucleic acid. You always have that alternating structure. This is yes, that will happen through dehydration reactions. All right, so phosphodiester bonds that attach these nucleotides together to form a nucleic acid, because of the way that they're attached, any nucleic acid has a sidedness to it. You're going to wind up with a free phosphate group at one end, and you're going to wind up with a free sugar group at the other end. We can label those groups, and each strand of nucleic acid has a sidedness to it. The 5' end is the side with the free phosphate group. 5' end is the side with the free phosphate group. That comes from how that phosphate group is attached to the sugar, the 5-carbon. Here at the other end, where you have a free sugar, is called the 3' end. So every strand of nucleic acid has a sidedness. You can identify the 5' end with a free phosphate group, and you can identify the 3' end with a free sugar group. Free hydroxyl, ready for that next to bond. So that's an individual strand of DNA or RNA. Now in RNA, it stays single-stranded. So this is what an RNA strand looks like. Here's ribose. This is what an RNA strand looks like. Alternating phosphate, sugar, phosphate, sugar, phosphate, sugar. You have the nitrogenous bases sort of hanging off on the side, giving RNA a sidedness to it, a 5' end on one side, a 3' end on the other. That's true for a single strand of DNA, but of course you know that DNA is found in the cell double-stranded. It is not found single-stranded. So when two strands of DNA come together, two strands of nucleic acid come together to form the double helix, those strands come together in a formation we call antiparallel. Antiparallel means the 5' end of one strand is next to the 3' end of the other strand. So they run in opposite orientations. They are antiparallel. I'm curious, how many of you have never heard any of this before? Okay, I just wanted to check.
I've never seen that diagram like that.
This diagram actually does come from your textbook. I cannot remember the chapter off the top of my head, but I can look it up for you. It's like 8 or something in that range, but I'll have to look. So yeah, DNA always has this sidedness to it, and the two strands come together to be antiparallel, where the 5' end of one is next to the 3' end of the other. The other thing, of course, you guys know this, that when they come together, that forms kind of a ladder structure where you have the sugar-phosphate backbones on each side. Right? You have the nitrogenous bases coming together, kind of like the rungs of a ladder. The nitrogenous bases come together. Also, you know that the nitrogenous bases don't just line up anywhere. They follow complementary base pair rules, which means that ATCG, which means that adenine always binds to thymine, right? And cytosine always binds to guanine. Right? So it's predictable. If you know what one strand of a piece of DNA looks like, you can identify what the complementary strand would look like. Let's do that as an example. So if I drew up, here's the 5' end, and it's right. So if here's one strand of DNA, if this is the 5' end, that means this would have to be the 3' end. And I expect you could identify what the other side of this strand would look like. It would be okay, and this end would be the 5', and this end would be the 3', right? So all double-stranded DNA is complementary, meaning the base pair rules are followed. It's also anti-parallel, indicating that opposite structure, where the 5' end of one strand is next to the 3' end of the other strand. An interesting thing to note, I don't know if we come back to it, but this is something that shows up periodically on standardized tests. It's a good thing to make sure I get out there. Notice the nitrogenous bases actually bond to one another through hydrogen bonding. That's how they attach to one another. And if you notice, adenine and thymine, when they line up, 2 bonds form between adenine and thymine. But when cytosine and guanine line up, 3 hydrogen bonds form between cytosine and guanine. So which one is the tighter base pairing? Cytosine and guanine base pair tighter together because 3 hydrogen bonds form between cytosine and guanine. With adenine and thymine, only 2 line up. So, what's that? Well, it's stronger because there's 3 hydrogen bonds. Yeah, I mean, each bond is the same, but the fact that there's 3 makes that interaction stronger. So if you had to predict an extremophile living in extreme heat, would you assume that it has more cytosine and guanines in its DNA structure or more adenines and thymines? Cytosines and guanines, and that's absolutely true. Those organisms that live in a really hot environment tend to have more cytosine and guanine in their DNA structure because that forms a tighter bond. And in fact, you can do all these cool studies where you raise the temperature a little bit and watch the denaturation of DNA, and you can anticipate its percentage of cytosine versus thymine, or those sorts of experiments. So it is a tighter interaction. I bring that up because I've seen it on standardized tests, and I know many of you have those in your future. So. Ah, see? All right. And then, of course, it is not just a ladder structure. Question?
So in cold environments, would there be more adenine and thymine?
Yeah, it's not as essential for those organisms, but yes, there are those who have more adenine and thymine levels. All right, so DNA double helix, of course, it's not just a ladder structure, but of course it has that twisted nature to it. It is twisted into a helix. I don't have it here, so I'm doing this from memory. It's an average of 3.4 nucleotides per turn. Does anybody know I'm right? I think it's 3.4. I'll double check it. It's an average of actually I can probably count on here in 2, 3. Yes, it looks like I'm right. 3.4 nucleotides per turn of the DNA double helix. I will double check that fact since I didn't write it down. Nucleotides per turn of the DNA double helix. I'll double check that data to make sure I'm not leading you astray. Now, DNA replication. When DNA replication starts, it always starts at a particular location in the DNA. That location is called the origin of replication, or ORI is short. Okay? It's a particular sequence in the DNA where replication always starts. So it's a predictable DNA sequence where replication will always start.
This isn't for eukaryotes.
I'm showing you a proc, and then I'm going to show you a euc next. Now, in a prokaryotic chromosome, right, we have a circular chromosome. Procs only have 1 ORI. They only need 1 ORI because when replication starts at that ORI, it does go in both directions. It's bidirectional. So here you can see replication at the ORI starts. Replication goes both directions at the same time. So you wind up with replication from both ends. What's the value of replication happening in both directions at the same time?
The internet says 10 nucleotides per turn, and it's 3.4 angstrom units.
All right. Thank you. So 10 nucleotides per turn, 3.4 angstroms is what I'm thinking of then. Per turn. Thank you. 10 nucleotides per turn, 3.4 angstroms is the space between turns. All right, so replication always starts at the ORI. What's the value? Why does replication go in both directions from the ORI? Speed, right? Absolutely, faster. Faster replication means that the cell can go through cell division faster. Important for a prokaryote. So bidirectional replication going in both directions at the same time. In fact, there's 4, because if you think about what's happening on each side here, you have replication happening on each side, and then on each side here. So there's actually 4 centers of replication happening. I'm going to come back and show you that in a minute. But when it's going in both directions and on both strands, you wind up with 4 centers of activity. Now, in a eukaryote, you have multiple ORIs on each chromosome. Now, it's not surprising to think that each individual linear chromosome is going to have its own ori, right? Because replication has to start on every chromosome, so at least one ori on each chromosome. But in fact, on most eukaryotic chromosomes, you have multiple oris on each chromosome. And again, the reason for this is speed. Because eukaryotes have introns and exons, their genome size is larger. You have to get through a lot of replication for each round of DNA, each round of cell division. So multiple oris allows that process to happen faster. And it is true from each ori, replication is bidirectional. Right? So just like we saw in the probe, from each ori, replication is happening in both directions. What will happen is eventually replication from this ori and this ori will meet up, right? And then it's completed that chromosome. It just allows it to go faster.
Are oris the same as Tata boxes?
No, the Tata box is involved in transcription. It's part of a promoter. So it's actually involved in a different process. That's part of the binding of RNA polymerase.
That's found on the mRNA?
No, it's found on the DNA, but it's a binding site for RNA polymerase for transcription. As opposed to replication, we're talking about DNA polymerases. So multiple replicons in the eukaryotic DNA. So to kind of go through the bits and pieces here, for DNA replication, of course, you always have your template strand that is being copied. It is semi-conservative. I'm going to show you more specifically what that means in a moment. But semi-conservative means that one strand is being replicated. Okay? The other strand is the template. So what you wind up with is every new piece of DNA is half old and half new. Probably went through that experiment in 1B. It's a classic experiment. But it's the idea that one strand is being copied, the other strand is new, so each new DNA strand is half old, half new. I'm going to show you a picture if that seems unclear. So hang out. I'll show you in just a second. At the replication origins, the origin of replication, or the ori, there's one single ori in E. coli, a prokaryote circular chromosome. That's true for all circular prokaryotic chromosomes. There's one single ori. There are hundreds in the yeast genome, at least one per chromosome, generally multiple per chromosome, are found in eukaryotes. There are thousands of oris in the human genome. Right? Multiple ones on each individual chromosome to allow speed. Again, the ori is a particular sequence in the DNA that attracts what are called initiator proteins. We're going to learn more about those in just a moment. That's what starts replication. So it's going to attract these proteins in that will start the replication process. If we look at the ori, it is actually quite easy to separate the two strands of the ori. They are not tightly associated together, and that's because they have lots of A's and T's in their sequence. That's true of the Tata box. It allows it to pull apart too. So A's and T's, of course, have weaker bonds than C's and G's. So therefore, it makes sense that this is a relatively weak area in the DNA, easier to pull apart and start the replication process. So the ori has lots of A's and T's in it, allowing that to occur, allowing those strands to get separated. DNA replication is bidirectional, happening in both directions at the same time from each individual ori. And it is a very rapid process. If we look for, at some numbers here, 50,000 nucleotide pairs per minute are replicated and made in bacteria. 50,000 per minute. I don't even know how to wrap my head around how fast that is. Right? They actually have videos. I'll link you to one. They have videos that show it in real time. It's amazing. It's a little cartoon that they've made, but they're trying to show it to you as fast as it really happens, and it boggles the mind to think that anything could go that fast. Attaching nucleotides together in order to make it happen extraordinarily quickly. In humans, it's about 2,000 nucleotides per minute are formed during human DNA replication. That's still really, really fast if you think about how fast that would be. All right. I mentioned it, and it turns out to be really important for cancer, so I want to come back and talk more about it. Here, I told you, we know the ORI is where replication starts. Why at the ORI? What's special about that sequence? That's what I want to talk about now. It's because this particular sequence of DNA is what's going to attract what we call initiator proteins. And the initiator proteins start the replication cycle. These are the proteins that actually say it is time to start replication. This is where we start. So here's the process. You guys are familiar with the cell cycle, right? Cell cycle, cell goes through a period of growth, DNA synthesis, another short growth period, and then mitosis occurs, right? Basic cell cycle. In fact, my kids learned that last week. They're doing biology apparently right now in 6th grade. We're going to come back to this because it's really important in cancer, so I'm going to go into a lot of detail there about cancer and this cell cycle and how it's controlled. But right now what we're talking about is the DNA synthesis phase, right? What's happening during this DNA replication phase. Now, it turns out that this entire cell cycle takes an average of 18 to 24 hours in a eukaryotic cell, but the length of time for any one cycle can be different. So we average them all out. But the length of time for a given cell is different depending on the kind of cell that it is. So what we really want to get to here is, how does DNA synthesis start? How does the cell know it's time to start DNA synthesis? What controls that process? What it's going to be is binding of these initiator proteins to the ORI. That's what's going to start the whole process. What starts DNA replication is binding of these particular proteins. So DNA in the nucleus is different in the mitochondria. Let's not worry about that for right now. We're going to worry about nucleus, right? Nuclear DNA is replicated once and only once during each cell division process. During each cell cycle, it's going to be replicated once and only once. Now, this is incredibly important. Why? Why can DNA only be replicated once during each cell cycle? Mutation's a possibility. What else? Those daughter cells, what's going to happen? They won't have the right number of chromosomes. If you have, you can wind up with all sorts of genetic problems. If you don't replicate just once to have double the amount of DNA in a normal cell before that cell division happens, you're going to wind up with the wrong number of chromosomes in the daughter cell. So this is a really important process for the cell to control. That DNA replication will happen once and only once with each cell cycle. So here's how this is controlled. They call it a licensing system. So the DNA gets licensed to replicate. Never occurred to me. That's kind of amusing, like license to drive. It's licensed to replicate. So it gets licensed to go through this replication process. And what happens is a number of proteins are going to come together, and only when all of those proteins are present together on the ORI is when replication can start. Right? So it's tightly controlled because there's multiple proteins that have to come in to set up that license to start replication. Now, if we look at the proteins that come together to form this complex that license the DNA, it includes ones that you've learned about before, helicase. Well, it's helicase loader. It's a protein that adds helicase. But you've also, helicase is one of what they call MCM proteins. Right? MCM stands for mini chromosome maintenance proteins. Let's just call them MCN proteins. That includes helicase. What is helicase?
Helicase helps separate.
It unwinds double-stranded DNA, pulls the two strands apart so that replication can start. Helicase is one of these proteins that's coming in during this licensing process. The first one in is called ORC. ORC is the origin recognition complex. Its job is to bind to the ORI, so it is always the first one in. So ORC is the first one in. ORC binds to the ORI. That's his job. It's very specific. He attaches to that ori. Next in, there's really two of them that kind of come in together. Helicase loaders do exactly what they sound like. They attach to ORC, and they allow helicase in. So helicase loaders are exactly what they sound like. They attach to the ORC protein, and they allow helicase to attach to them. So our sequence here is ORC binds to the ori. Of course, ORC is a protein binding to the ori, which is a sequence in the DNA. Next, really together are the helicase loaders. And helicase. They're going to bind to ORC. Now, wouldn't it be easier if it was just one protein that attached to the DNA? Why in the world is this complex thing evolved where there's 3 different kinds of proteins that have to bind to the ori? Why is that beneficial? Because of course it will only evolve if it was beneficial. Regulation. Remember, it's incredibly important that replication happens once and only once. So the cell can use these proteins and then immediately degrade only one of them and stop any future replication. So it allows multiple levels of control. The cell can get rid of any one of those proteins and stop replication when it wants to. So there is value in having multiple proteins involved in the process. You have multiple ways to control the process. And this process is incredibly important. It has to be controlled. You cannot have multiple copies of DNA in your daughter cells. So multiple levels of control are the value here. So ORC in first, and then the helicase loaders and helicase come in together. Once they have all attached together at the ori, now the DNA has officially been licensed to divide. It now can go through cell division. Couldn't go through cell division until all of those initiator proteins attached. All right. Now, once those proteins have attached, the replication cycle is really, or DNA replication is really carried out by what we call polymerases, DNA polymerases. DNA polymerases are the actual enzymes that read one strand of DNA and they synthesize a complementary anti-parallel other strand. Walk through that as I said that, right? So a DNA polymerase will read one strand of DNA and synthesize a complementary anti-parallel other strand. With me? Right, complementary means using complementary base pair rules. Anti-parallel means it has to go in the opposite orientation. So DNA polymerases form that bond, that phosphodiester bond between the 3' end of one strand and an incoming 5' end of the next. Right, so they bring in nucleotides to build that strand. During the process, 2 phosphate groups are lost from our nucleotide, releasing pyrophosphate. These 2 phosphate groups together get released during this process. So DNA polymerases read the template strand, synthesize a complementary anti-parallel strand. Now, notice, I think I saved this. Oh, I have it on the next slide here. Interesting, DNA polymerases are valuable in their job for sure. They do the actual replication of DNA, but they have limitations. They have interesting limitations. They're sort of meaty enzymes. The first off is they can only add nucleotides to the 3' end of the strand. So DNA polymerases only add nucleotides to the 3' end of the strand, which means all new DNA is made in the 5' to 3' direction. So if I back up here and show you that picture, DNA polymerases can only add to the 3' end. They're always growing the 3' end. They cannot add to the other side. That's the only side that they can attach to. That means all new DNA is always made by extending the 3' end. In other words, it's made 5' end first, 3' end last. So all new DNA in any cell of the world, all new DNA is made 5' to 3' because of this limitation of all DNA polymerases. They all have that limit. Now, this is going to allow an extra ability. We'll see that most polymerases can also proofread, which is a value. They can tell if they did something wrong. They'll pull off an incorrect nucleotide. I'll show you that in a few minutes here. But that is going to result in the leading and lagging strands that you've seen before during DNA replication. The leading and lagging side of that replication comes from the fact that DNA polymerases only add to one side. The other limitation for DNA polymerases is that they can't start replicating without something to attach to. They can't just grab on and start copying. They have to attach to something. We call that something a primer. So DNA polymerases require a primer in order to go through replication. I always think of it as they can't attach to the DNA unless they have something to attach to. They attach to that RNA primer. That requires an enzyme to make the primer. That enzyme is called primase. So primase must make that short RNA primer. Unfortunately, since the RNA primer is RNA, that primer also has to be removed at the end of replication and filled in with DNA, causing an extra step to happen. Let's go through those steps here. So here, if we start out, we are looking at, in dark blue, we're looking at complementary base paired anti-parallel DNA. During replication, replication is happening actually on both sides at the same time. Both sides of that template is opened up for replication. On one strand, replication is happening in a leading fashion. In other words, as the dark blue strand opens up, here's the 3' end of this strand. The new strand is going to be anti-parallel, so you know the new strand has to be 5 here. Yes? So what happens is DNA polymerase is going to just sort of follow along. As this strand opens, it's going to keep copying, keep copying, keep copying. Why can it do that? Because remember, it goes 5 to 3. So DNA polymerase is happy. It's copying as this strand continues to open, it continues to attach and continues to copy. That's the leading strand. So in the leading strand, replication happens replication is continuous by polymerase is continuous in the 5' to 3' direction. On the other strand, the dark blue template has opened up here. Our parental dark blue DNA, 5' end on this strand. So we know that the new DNA has to have 3' end on that side. So can DNA polymerase just attach and copy this direction? No, why not? It always makes DNA 5' to 3'. So if it tried to do it this way, that would be the wrong direction, 3 to 5. It can't do that. Right? It always makes 5 to 3. So instead, what happens is, as that strand opens up, DNA polymerase will attach, make a short piece of DNA. Then as the strand opens a little bit more, it will attach, make another short piece. As it attaches, as it opens some more, it makes another short piece. So it makes the DNA in these discontinuous segments, these short segments called Okazaki fragments, after the guy who discovered them. So on the lagging strand, replication by polymerase is in short segments. But each segment is 5 to 3. It's always done 5 to 3. So because of DNA polymerase's limitation of only adding to one side, you wind up with this leading strand on one side and lagging strand on the other side.
What's the protein that binds the Okazaki fragments called?
Anybody remember it? We are going to talk about it in a few minutes. What protein binds Okazaki fragments together? Ligase. Ligase. All right, so while we're here, another thing that went by, but I want to remind you of because I got some kind of confused looks. Remember I told you that DNA replication is semi-conservative? What semi-conservative means doesn't mean you sort of kind of like Mitt Romney. That's not what semi-conservative means. Semi-conservative means that each new strand of DNA is half old, half new. Right? So each new strand of DNA is half old, half new. So if you're looking at this strand here, the new strand is half dark blue parental and half brand new light blue. Right? So it's here. So half of it's the old parental, half of it is the new. That's what semi-conservative means. So what's that?
So half of it is the template.
Right. Half is the template, half is what was replicated. That's what semi-conservative means. When you hear that, it just means that it's half old, half new. It seems obvious to us now that we understand the process of DNA replication. It wasn't so obvious when they were first discovering replication. All right. So another way to look at this here, remember that at the ORI, if we kind of walk through the steps here, at our original ORI here, first in was to license. Who was the first to attach to the ORI? Still over there. ORC binds to the ORI. He brings in helicase and helicase loaders to license the DNA for replication to occur. When DNA polymerase gets in, remember it's opened up, helicase has opened up our double-stranded helix here. Replication happens from this ORI in both directions at the same time, which really kind of gives us 4 centers of activity, because if you're going this direction, you're going to have a leading and a lagging strand. And if you're going this direction, you're going to have a leading and a lagging strand. So you really kind of wind up with 4 areas of replication happening all at the same time. That's what this is showing you. They're showing you as replication moves away bidirectionally from that ORI, you wind up with replication happening here on a leading strand. Parental DNA, here's 5', which means a new one is being made. You could label it here 5' to 3'. And on this side, it's happening 5' to 3'. Question? Yeah, helicase unwinds the DNA. Polymerase copies the DNA. And we're going to go through step by step. Actually, in a couple of slides, I'll actually walk you through each step in the order of the steps. Leading, lagging, okay? Right, centers of replication. All right. Now, I mentioned this, but I want to come back to it. The fact that many DNA polymerases have proofreading ability as well. Proofreading means that not only can they attach nucleotides together, but they can check to see if they made a mistake. Most polymerases have that ability. So they kind of have several abilities here. DNA polymerases can add nucleotides to the 3' end during replication. That's what we just talked about. They can add in those nucleotides. They can cleave pyrophosphate from that nucleotide as they attach it. Right? So as they attach that one, they release pyrophosphate along the way. The other thing that's really important is that they can proofread. They can actually cut off the last nucleotide if they see there's a problem. So if it recognizes that it added the wrong nucleotide, it can cut that one off. So it has what we call exonuclease activity. It can cut off a nucleotide from the end that it just added it onto, so from that 3' end. So here's an example here. DNA polymerase reading the template strand in dark blue, synthesizing the daughter strand in light blue. It's reading here, it sees that there's an A, so of course it needs a nucleotide that has a T, so it should select that nucleotide and add it on to the growing 3' end. That's what they're supposed to do. It sees a T, it adds an A, etc. Here it sees a T, but somehow by accident it's adding a G. Turns out it also has the ability to cut that G back off. So if it recognizes the problem, it will cut it off of that new end. They have this proofreading ability, the ability to self-check themselves as they add on to the DNA strand. So most polymerases have proofreading ability as a 3 to 5 exonuclease, so they can cut off the 3' end if it's incorrect. They can't go further back. They can cut off 1, sometimes 2 nucleotides, but that's it. They can't go back to any strand, but they can cut off just the end. That actually reduces the error rate in new DNA. So this becomes important in preventing mutations. DNA polymerase is doing its job very, very quickly, copying, making new DNA very, very quickly. If it accidentally adds in a wrong nucleotide, it can pull that out, making a very low error rate. 1 error for every 10 to the 7th nucleotide pairs it copies. That's incredibly good if you think about it, right? 1 error in 10 to the 7th nucleotide pairs that it reads and produces. That's really amazing. Better than any company's error rate, I'll tell you that. All right, so most of them have the ability to do this little checking of the DNA as it goes through. Now, my experience is from this point, you guys have heard about DNA polymerase 1 versus DNA polymerase 3. Yes? You learned about this in prokaryotes. So DNA polymerase 3 is the one that does the major copying in a prokaryote. DNA polymerase 1 has a job later on in a prokaryote replication process. I want to talk about Ukes, however. It's very similar, however, they're not named 3 and 1, like you have learned before. Instead, the major DNA polymerases in a eukaryote are DNA polymerase alpha and DNA polymerase delta and epsilon. These are the ones that are involved in nuclear DNA replication. A little bit different in the mitochondria, but these are the ones involved in the nuclear replication. DNA polymerase alpha. DNA polymerase alpha is the main enzyme for initiating DNA replication. He always starts replication. This is the one that attaches to a primer and starts the process. He's always first polymerase in. He always begins replication. That's true on both the leading and the lagging strands. So DNA polymerase alpha is the initiator polymerase. He is going to add to the primer, he attaches to the primer, and then begins to extend that process out. I'm going to show you a picture in a moment. This is just sort of background on what he does. Now, the value here is he's very good at attaching to a primer and starting replication. He's actually more efficient at it than the other polymerases. The downside is he doesn't show proofreading ability. He's an exception to our rule. He doesn't have proofreading. So you don't want him to be around for very long. You want him to start replication and get out of the way so somebody who can check their work can get in there. So he's very good at starting, but then you want him out, and you want to allow the other polymerases in. So DNA polymerase alpha starts replication, but because he has no proofreading ability, he gets kicked off and replaced with one of the other polymerases later. We're going to see that DNA polymerase alpha is actually attached to primase. They are complex together in a eukaryote. The one that makes the primer attaches to alpha so that they can start replication right away. Now, if we look at delta and epsilon here, DNA polymerase delta and epsilon are both thought to be involved in synthesizing leading and lagging strands. So they're the replacements for alpha. Alpha starts, alpha gets out of the way, and allows delta and epsilon in. They both have proofreading ability. They both can do their exonuclease, cut out anything that was wrong. It appears that DNA polymerase delta also replaces the primer with DNA after it is digested away. So this was the job done by DNA pol 1 in prokaryotes. You may remember that DNA pol 1 cuts out a primer and fills in with new DNA. The one that's doing it in eukaryotes is delta. It's DNA pol delta. Let's let's walk through the steps. All right, so the process here. Primase. So if we look at the actual replication process, in comes primase, the enzyme that is going to start replication by creating an RNA primer. Why do we need an RNA primer?
Start the attention.
Remember, one of the limitations of polymerases is, one, they only add to the which end? To the 3' end. And the other limitation is they can't attach without a primer. That creates the need for this extra enzyme primase. In a eukaryote, primase is attached to DNA pol alpha. So primase comes in, he makes that little short primer, a place for alpha to attach to. Alpha will attach to that and starts DNA synthesis in the 5' to 3' direction. Pretty quickly into the process, DNA pol alpha will get kicked off. In will come delta or epsilon. They will attach and continue that process. So DNA pol alpha adds to the complementary DNA to make a short strand, then delta and epsilon take over replication. Eventually, at the end, you're going to have to get rid of that primer for complete replication because the primer is RNA. You may remember in a prokaryote that's done by DNA pol 1. Here it's actually done by an enzyme called RNase H that removes the primer, and it's filled in by DNA pol delta, fills in with new DNA. Any little nicks or holes that are left over are going to be sealed up by his favorite, ligase. Ligase will seal any pieces together. Check and see. All right, I want to add one more enzyme into the mix, and then we're going to do the whole thing one step at a time, show the process here. The other one I want to add in, we've talked about helicase as being an unwinding enzyme. But that unwinding actually creates a bit of a problem. Unwinding creates tension in the DNA strand. If you think about a double helix that's coiled together, if you open it up here, it's going to cause supercoiling on each side, right? It actually winds the strands tighter together, which creates tension and possible stress on the DNA. So there is an enzyme to take care of that called topoisomerase. Topoisomerase gets rid of supercoiling. Topoisomerase releases supercoiling in the helix during replication. It's kind of a funny process. What it does is it will actually cut through one of the strands, allow it the tension to uncoil it, and then it will seal it back together. It's kind of a cool thing. So it actually creates a little nick on one side. The strand rotates around till the tension is gone, and then it reattaches it together. So topoisomerase gets rid of this tension from supercoiling. That happens during replication. They're showing it as being right on top of the replication bubble, but in fact it's quite a bit back in the DNA because the tension forms further back from that supercoiling, and that's what topoisomerase gets rid of. The other one we haven't talked about here, the single-stranded DNA binding proteins. Those are there. Remember, DNA wants to be double-stranded. Those hydrogen bonds will form to keep it together. Helicase is unwinding. The single-stranded binding proteins keep it unwound. So they prevent recoiling of the DNA before replication happens. Single-stranded binding proteins. Single-stranded binding proteins prevent the helix from reforming. So hold the two strands apart during replication. We're going to hold those two strands apart during replication. All right. let's put the whole show together from start to finish. All right, to start out with, before any replication can occur, the DNA has to be licensed. Right? That is definitely the take-home message. DNA has to be licensed, tightly regulated process. So licensing, of course, first guy in is ORC. ORC is a protein that, what does he do?
This applies to prokaryotic.
This is eukaryotes, or I'm going to specifically talk about eucs now. So ORC binds to the ORI. His whole job in life is to bind to the ORI. ORI tends to be AT-rich, which is going to allow it to open up and allow replication to happen. Once ORC has bound, it allows in helicase loaders and the helicase enzymes to come in. All of that together creates the license to go through replication. That will be tightly regulated in the cell cycle. We'll learn more about how it's regulated later. But that's all tightly regulated to ensure one replication happens per cell division process. All right, so all of that has formed together. That's allowing in helicase. Here, next we have unwinding. Helicase is opening up the strands. Single-stranded binding proteins are holding the strands apart so that they don't spontaneously go back together, because they want to spontaneously go back together. Topoisomerase is reducing supercoiling. That's happening other places in the DNA. Creates those nicks to relax supercoiling that could happen in the DNA. Kind of going fast, but I feel like you're all with me. Yes? All right, so we got helicase in to unwind, single-stranded binding proteins to hold it apart, topoisomerase to get rid of supercoiling. Next step, priming. We got to create a primer for polymerases to attach to. Primase is actually attached to DNA full alpha in a eukaryote. There's one complex that's together. Primase comes in and it makes a short RNA primer. They're showing it on one strand, but of course it would be happening on both strands. So you're going to have a primer being made on both strands. Once that primer is made, our first DNA polymerase to attach is, well, he's already there, DNA pol alpha. DNA pol alpha attaches to that primer, starts the replication of DNA. Does he last long? No. Why don't we want him to last long? No proofreading ability. So he starts the process, he gets kicked off, in comes delta and epsilon to continue the process. Continue that process during replication. Now this is happening on both strands at the same time. They're showing you the leading strand first. So here's our primer that was formed. DNA polymerases attach and continue that process in the 5' to 3' direction. They're showing you the leading strand. Of course, this is happening on the other side as well, on the lagging strand. On the lagging strand, you have a primer that was made. Pol alpha is attached. He gets kicked off. Delta and epsilon come in to continue that process, also in the 5' to 3' direction, but in short segments. Now what's going to happen here is primase will have to make a second primer, and then a third primer, and then a fourth primer during that replication process. Yeah, so you will have, and in fact, when we teach it, we show them as discrete individual entities, but in fact what's usually happening is you have a giant mass of enzymes all attached one to another, and so they just sort of take over for one another. So it's not quite so kind of, your turn, I'll get out of the way, the next guy's turn. They're kind of one mass of enzymes, and as the DNA goes through, each one does its job. The way they always show this is that the enzymes are moving, but if you think about it, enzymes are great big giant proteins, while the DNA is tiny. So what's actually happening is the DNA is getting fed through the enzymes, because the DNA is much smaller. We always show it this way because it's easier to conceptualize, but in reality there's a giant mass of enzymes and the DNA gets fed through that mass of enzymes. All right, so lagging strand, you have multiple primers. DNA pol alpha starts after each primer. Delta and epsilon takes over after each primer. Next, we're going to have to remove the primers. RNase H is the enzyme that does that in eukaryotes. RNase degrades RNA. That's right. RNase H is degrading out that RNA. And then we have DNA pol delta filling in with new DNA here. Once, of course, on our leading strand, multiple times on our lagging strand. Each individual primer has to be gotten rid of and then filled in with new DNA through that complex. The last step is to seal together any little pieces that might need to be sealed together, and that, of course, is ligase. Ligase will seal together the bits that are left over from Okazaki fragments and anywhere else. Of course, remember this is happening from multiple ori, so eventually this leading strand will meet up with another strand and it will need to get sealed through ligase as well. Any nicks that are left over after the process will be fixed by ligase. Questions? So we kind of did each individual one and then put them together in order. It's a very similar process to what you no doubt learned in Prox. The difference is it's a little bit more complex because there's multiple different polymerases, but it's the same overall process. Now, what I sort of mentioned a moment ago, I'm going to show you a picture of, it's always boggled my mind to think about these individual centers of activity and how they're happening. But in a cell, of course, enzymes are much larger than the DNA. So the enzymes are actually static. They don't move the way that the DNA does. What they're showing you here is there's quite a bit of evidence to show that the lagging strand and the leading strand enzymes are actually the same, or at least they're in one large complex together. So you'll have a giant complex of enzymes that includes multiple copies of primase and multiple copies of DNA pol alpha, multiple copies of delta and epsilon. You'll have RNase H, all of them in together, forming kind of this giant mass of enzymes that the DNA then gets cycled through during replication because enzymes are heavier than DNA. In order to allow that to happen, the lagging strand generally is bent and turned around so that one giant mass of enzymes can do both at the same time. This is efficiency, because if we think about three-dimensional space, you just coil that around and now everything's going the same direction. It allows the process to happen faster. So lagging strand DNA is actually folded into a loop, allowing DNA polymerase molecules on the leading and lagging strands to come together and move in the same physical direction. It's really not them moving, it's really the DNA moving, but it's holding them all in one area together, allowing that process to happen more efficiently. This is a table that comes from your textbook showing you all the different polymerases. I have told you the ones that I want you to know about, but should you want to go back and look at the details of any one enzyme, they are here in your tables. It's table 19.1 from your textbook. They show you exactly what's happening in bacteria versus what's happening in the nucleus versus what's happening in the mitochondria. We're focusing on nuclear eukaryotic replication because that's what we're going to be concerned with cancer. But it does go through all of that. Talks about what single-stranded binding proteins do, how topoisomerase works, all of that stuff is here in this table. This is actually a good place to stop. So I am going to pick up here on Thursday. Finish up, there's just a last little bit here, and then move into the next topic of cancer. We're going to talk more about cell cycle control. Now that you know about licensing, we'll talk more about how that cell cycle is controlled and what can happen in a cancerous cell. So let's take a quick break, and then I'll come back for lab. I actually am going to have to go. Let me stop this recording. You don't need all of this in recording.